So Jason, Holliday is our next speaker. Take
Thanks.
it away.
Looks like I've only got 3 minutes until the next thing, so I'll try to be quick. Okay, so you already had a nice background from Jared on American chestnut breeding and restoration efforts. I'm going to focus in on one narrow aspect of that, which is our efforts to understand natural genetic diversity in the wild in those stump sprouts that remain for chestnut, and to try to get towards a strategy for conserving that germplasm. And this is mostly work by a PhD student, Alex Sandercock, who couldn't be here. He's actually doing an internship this summer at a company. So our ultimate objective, as I mentioned, or as Jared talked about briefly, is we want to increase the effective population size and the adaptability of those, especially the transgenic chestnuts, the blight-resistant transgenic chestnuts. So through crosses with wild pollen. And so how do we get there? What we decided to do was to use a whole genome sequencing approach to understand neutral and adaptive diversity in that sort of remnant populations of chestnut. So we over a couple of years, we sampled 384 of these wild stump sprouts of American chestnut. A few of those were large surviving trees. We also sequenced 95 Castanea congeners, so all of the other Castanea species at about 20x. Actually, it turned out to be about 17x. And so this is just illustrating all of our sampling sites. The blue triangles are the dentata, the American chestnut, and then some of the sympatric congeners, Castanea pumila, and a couple of subspecies, alabamensis and ozarkensis, which are these three sort of recognized groups of chinkapin in North America. So why do we need whole genome sequencing to effectively sample this wild adaptive diversity? Well, my hammer happens to be genomics, but there's a more important reason, which is that these stump sprouts in the wild hardly ever produce nuts, and so we can't do conventional provenance tests in order to understand the distribution of adaptive traits across the range. And so we did all the sequencing, and the first thing that we did was to try to understand whether any of these sprouts that we sampled were hybrids. So don't worry too much about the coloring of all the dentata samples. I'll get to that next. These are the congeners, and the question is, were any of these quote unquote wild samples, are they hybrids? Because there's a long history of planting these exotic species in the US. In general, no, we did find a few sativa hybrids and a couple of Melissima hybrids, so Chinese chestnut, and a couple samples that were actually looked like they're chinkapins. A question for a later date is, what is the nature of these three chinkapin subspecies, alabamensis, which is in northern Alabama, ozarkensis, which is in Arkansas and Missouri, and pumila, which is the typical chinkapin of the Appalachians. And so it looks like ozarkensis at least has some contribution from one of the dentata populations, so there might be some introgression or something going on there. But we're going to be looking more deeply at the comparative side of this with some other analyses, not that I won't talk about today. So what did Alex find when he used admixture? He finds that we can roughly divide Castanea dentata American chestnut into three populations: a relatively homogeneous northeastern group, a central group that encompasses the sort of center of diversity of the species, so along the spine of the Appalachians, and then this distinct southwestern population in the warmest part of the range. Interestingly, if he allows for another cluster, what we get is a separation along the spine of the Appalachians, such that the east and the west, or the east and the west side of the Appalachians become sort of two separate populations. And so that may indicate the history of post-glacial migration up and down, or up either side of the Appalachians as they moved out of their glacial refuge. So like many tree species, chestnut has experienced periods of decline and rebound through the Pleistocene. So going back 2 million years, we see this decline in effective population size, and then sort of up and down as the glacial cycles sort of ebbed and flowed over the last couple million years. Since the most recent glacial maximum here, the effective population size has rebounded. Interestingly, so this is that southwestern group ends up having the largest effective population size. The central group, the northeast group is second greatest. We have this little dip in the central group, which might be just a statistical kind of artifact or something. That we don't expect to see a decline as a result of the blight-induced bottleneck, because there has been no sort of subsequent drift and evolution of the species, because there's no generational turnover. We see clinal variation in nucleotide diversity and the frequency spectrum. So the southwestern population on the bottom here, the bottom panel here, has the highest diversity in spite of being sort of the geographically most constrained and smallest population. So that's indicative of it being a glacial refuge and a melting pot of diversity over a long period of time. And by contrast, the orange line here is the northeast group, and it has the highest diversity. If we look at the frequency spectrum summarized with Tajima's D, we see that the southwest population has the most negative sort of average genome-wide Tajima's D, and the northeast has the closest to zero. And this is indicative of the history of bottlenecks, with the southwest being the most ancient population bottleneck, and the northeast being the most recent. And the reason I say that is because if we look at the distribution of the allele frequency spectrum across millions of alleles, what we find is that for the bin that encompasses low frequency alleles, the northeast population has a relative deficiency of low frequency alleles. And that's what you would expect if there was a recent sort of truncation of the gene trees as a result of a population bottleneck. So southwest, relatively stable long-term effective population size that has a wealth of both rare and common diversity. In the northeast, a recent bottleneck associated with post-glacial recolonization. One of the ways that Alex started looking at natural selection was to use this raised D statistic, which is kind of a composite of a variety of different statistical tests to look at the history of natural selection. I won't talk too much about this because I've already talked too long probably, but one of the interesting things that come out of this is that among all of the selection outliers for the three different populations, the thing that is most significant in terms of gene ontology terms is defense response to fungus. So this doesn't likely indicate that that genes related to, say, chestnut blight have been targets of selection, but there is a history of selection related to fungal pathogens across the chestnut range. And these genes might be interesting to look at in the context of blight resistance, in spite of the fact that we're not saying that they are sort of direct targets of the blight. And so one of the primary goals of this work is to sort of circumscribe adaptive units across the range and decide how to sample that diversity for the purposes of breeding. And so Alex did a redundancy analysis to try to look at the multivariate genomic space that comprises adaptation to environmental variables in the species. And so we find that the greatest contribution comes from temperature and precipitation variables in terms of selective constraint. And the second most significant is evaporative demand. So the combination of heat and moisture in the warm parts of the range appear to be an important driver of adaptation in chestnut. I don't have time, obviously, to explain how all this works, but we can talk more about it later if you like. In terms of how climate change can be expected to affect this species, now the samples that are on the ground we don't care about because they're all just stump sprouts. They're not going to adapt. They're not going to further evolve. But we do care about how we deploy this natural diversity on the landscape in the future from the crosses with the blight-resistant breeding samples. And it looks like, as you might expect, in the Northeast especially, we're going to see a significant mismatch between what is the diversity that's presently there and the environmental conditions in the future. So ultimately, our goal is to vegetatively propagate a large number of samples from across this historical range on the basis of dividing the range probably into 3 sort of management units that are defined by the neutral population structure, and then within those management units trying to subsample them into relatively homogeneous groups with respect to their adaptive allelic content. So yeah. This is obviously work of a large number of folks that help with sampling, with the lab work, and developing that reference genome that was invaluable for doing this kind of analysis. So thanks.
Have you done demographic features reconstruction to know if the effective size has maintained a long time?
Yeah, so that's what. So I kind of glossed over that, but that's what this one would be. So this is the SMC++ analysis, looking at going backwards in time and the effective population size. And so this is so far back I wouldn't even worry too much about it. But you know, at the end of the glacial period in gray here, the population size goes up. At the start of this glacial period, it goes down, and then at the end of the most recent one, we see the population sizes going up again. Yeah. Sorry, I was going way too quickly through all of this. So yeah.
How did come to the conclusion of crossing the pandemic?
This is based on simulations that Jared did to try to get the effective population size up to 500. And so maybe you want to explain that a little bit.
Yeah, just based on inbreeding the spec from doing intercrossing, after doing that outcrossing from wild trees, and then the effect population size. That I was just trying to figure out what's the minimum amount of effort we need to do to sample that wild population. And I think this adds another dimension to it, but it's still like figuring out the efficiency of like, are we capturing all the gap in space with our collection of important tests?
Yeah, seems like you have a very clear case for assisted migration for parental selection and breeding.
Yeah.
You could use those southwestern alleles.
Yeah, and I think that, and so I skipped over a lot of that, but yeah, I mean, I think that capturing, we're going to have to do some disproportionate sampling in the Southwest, because even though it's a small part of the range, geographically speaking, it's a wealth of diversity down there, and it's diversity that's going to be important, as you say, in the going into the future. It's the diversity that's related to adaptation to, you know, warm, high temperatures in the summer, and also the interaction between temperature and water, which I think is really important. Yeah.
But from an adaptation standpoint, temperature and moisture, how far geographically would you need to go right now for 20 years out, 50 years out?
How far would you have to move a sample? I don't know about the exact numbers, but well, so yeah, I mean, I don't know if I could tell you like on a kilometer basis right now, but yeah.
So if you were certainly southwest, alleles would be useful in the center.
Yeah, definitely. I don't think you're moving alleles from the Southwest up into Maine or something like that, of course, but and it's complicated because the Appalachians, funny things happen in the Appalachians because you have, this high elevation, more northern-like environment. And so like, it depends on exactly, what your elevation is in addition to what your latitude is.
Okay, Jason, thanks so much.